Let's begin. Thank you everyone for joining us today for our Friday noon talk. Today we have the pleasure of welcoming Dr. Ravi Menon. He's a professor of medical biophysics, medical imaging, and psychiatry at Western University, where he's also a member of the graduate program in neuroscience and the graduate program in biomedical engineering and scientific director of Western Center for Functional and Metabolic Mapping, Canada's national ultra-high field MRI facility. In addition to this, he's also a fellow of the Royal Society of Canada. He's also a senior fellow of the International Society for Magnetic Resonance in Medicine and the Canadian Academy of Health Sciences. He was twice an awardee of Canada's Top 40 Under 40 program and a recipient of the Explorers Medal from the Canadian Institute for Advanced Research. And as well, he is, like many of our speakers this year, a graduate of the MPU program. As I understand, he was one of the first under the Podgorsak era of medical physics here at McGill. So we're very happy to have him here today to speak to us about his journey from physics to functional brain imaging. So please take it away,
Ravi. Thanks very much, Pete, and thank you all for coming to this talk. I think this is sort of dedicated to the students in the MPU, so we're going to have some fun. We're going to learn a little bit about functional imaging, a little bit about physiology, and a little bit about physics. This will be, I think, pretty different than from your typical MPU course load. I thought, this is actually quite interesting because, it's been 35 years since I graduated from the program, and I've probably spoken at least a dozen times at McGill, I'd say, in that time, talks and named lectureships. But this is coming home. I've never actually spoken at the MPU or visited the MPU since I left, and so this is actually quite fun. And so you'll hear a little bit about the history of that too. So, as I said, this is going to be for the students. So the take-home message for the students, as you'll see in my life, is that your path can actually be very convoluted. Most people think, success, however you choose to define that, is kind of this linear thing. You set your mind to something and you work hard and you achieve it. But that's actually not the way it works at all, at least not in my experience. A lot of luck is also involved, typically being in the right place at the right time and recognizing the fact that you're in the right place. You can tip the odds in your favor by, I think, loving what you do, being passionate about it, and working really hard at it. So I'm going to talk to you a lot about the discovery of functional MRI in the first part of my talk. And that kind of holds, true to what I just showed you. A lot of things have to line up in just the right way for a big discovery to happen. So that's certainly true for functional MRI and the role that I played in it. So let's kind of review that. So the story of functional MRI starts with a guy named Linus Pauling. Many of you will know. Linus Pauling won 2 Nobel Prizes. Many people don't know that, one for peace and one for chemistry. We'll come back to him later. These 2 gentlemen, Ed Purcell on the left and Felix Bloch on the right, were at Harvard and Stanford in the 1940s. And they discovered the principles of nuclear magnetic resonance. Harvard was very snooty in those days and didn't believe in patenting things, but Stanford was kind of the Wild West, and so they did file patents, which they then sold to a company many of you will know very well, Varian. So the 2 Varian brothers started a company that initially built klystron tubes, but later in the 1950s started building NMR machines, and of course linear accelerators and things like that later on. So using the principles of NMR, Linus Pauling hired a guy named Hardin McConnell when he was at Caltech to be a young assistant professor. And McConnell was a biophysicist who used NMR to study the structure of molecules. The Varian brothers, in the meantime, hired a guy named Richard Ernst, who worked there developing what became known as multinuclear NMR, or two-dimensional NMR. And one of his seminal papers with a guy named Kumar was actually the technique that we use for MRI reconstruction to this day. He used the payroll computer at Varian to actually do the Fourier transforms because there were no computers powerful enough to do things like that. McConnell, meantime, left Caltech and he went to Stanford in the early 1960s. And he hired a graduate student by the name of Seiji Ogawa. Seiji Ogawa used NMR to study the structure of hemoglobin, which was a molecule that Linus Pauling had something to say about, as I'll tell you later. So based on some of the principles that Ernst developed in the 1960s and early 1970s, these two guys, Paul Lauterbur and Sir Peter Mansfield, independently discovered or invented magnetic resonance imaging. Lauterbur using a Varian A60 NMR machine, and Mansfield using a slightly different apparatus. Now, the interesting thing about Purcell was he was something of a polymath. So Purcell's first graduate student in the NMR area was a guy named Charlie Slichter. And Slichter wrote a very famous book on NMR that I and many other people have on their shelves. And he went to the University of Chicago later on. But Purcell got bored with NMR very quickly, and he invented a whole other field called radio astronomy. And his first graduate student in that area was this guy, who turned out to be my dad. He met my mom, and I was born in the 1960s. But going back to Slichter, Slichter moved. He went to the University of Chicago at Illinois. And he had a graduate student by the name of Meyer Bloom. Meyer Bloom did his degree in NMR as well. And then he moved to the University of British Columbia in the 1960s. And he stayed there for his entire career. We'll come back to him as well. Now, the interesting thing about Mansfield, rest in peace, was he was kind of a hard ass, to put it mildly. And he had a young junior faculty member at the time by the name of Peter Allen. Peter Allen was a brilliant NMR spectroscopist, very interested in vivo work, but didn't, like many people, get along with Peter Mansfield. So he left and he moved to Edmonton, Alberta, or he set up Canada's first, real NMR MRI research center. He did that in the early to mid 80s. Mansfield was knighted in 1993, and then both of these guys won the Nobel Prize in 2003. Seiji Ogawa, after he got his degree from Stanford, he moved to Bell Labs and set up the biological sciences and biological physics division there. And he had many famous postdocs, Ian Smith, who became director of the National Research Council here in Canada, Camille Ugerbil, who ended up becoming my postdoctoral supervisor at the University of Minnesota, Bob Schulman, who became the director of the MRI Center at Yale, and many, many other stellar people over the years. So all of this, starting back in 1936, kind of fortuitously came together to when I moved to Minnesota as a postdoc and was involved in the discovery and the development of functional MRI. So we'll trace out bits more of that as we go along this path. So how did I get to McGill and how did I get to the NPU? Well, I started at University Hill Secondary in Vancouver, which had nothing to do with the university. It was just on a hill next to the university. And I started at UBC in the physics honors program. And that was a very, very tough program, actually. And the first couple of years, I didn't do brilliantly, I would say. This was considered, along with Caltech and MIT, to be the 3 top physics programs on this continent. And then I joined the motorcycle club because that's what, rowdy people do. Also, parking was $1 a year for a motorcycle. So that was a good way to get around in Vancouver. And that was the physics building, the Hennings building, which pretty much looks the same even now. But in third year in quantum mechanics, I met Meyer Bloom. Remember Meyer Bloom from Charlie Slichter's grad student? So Meyer was a very distinguished physics professor, very well known in the NMR field. He was a fellow of the Royal Society of Canada already, even by that time. And he piqued my interest in biophysics and medical physics and NMR. And he became something of a mentor to me. His alma mater was McGill, and he suggested maybe I might like to go to McGill to learn something about NMR and MRI. But I wasn't really sure. I think many of you, was motivated to apply physics to problems in health and to benefit, clinical practice in some way. I didn't have a really good sense of the concept of medical physics or anything. But, they were doing positron emission tomography work at Triumph, the big cyclotron in Vancouver. And so I thought, hey, you know what? Let me apply for a job as a cyclotron operator at Triumph. So I graduated from physics, I applied for the job, I got the job. But the director told me I'd be really bored running a cyclotron, and I should go to grad school. So I thought about Meyer's comments about McGill, and I applied, decided to go to McGill, but I didn't have a scholarship to go to McGill. I just had savings from 4 years of summer jobs at UBC in various research labs. So, there were some bumps along the way, but I bought a truck for $800, and a friend of mine, Alan Sorensen, who also graduated from the same program, we drove across the country. He went to McMaster in radiation physics there, and I drove further up the highway and went to McGill. And that sounds so easy, but actually, as I said, there were a few bumps in the road, and I dug this up just last week. So this was a letter. So people used letters in those days, and they were mailed with a stamp. There was no email, of course. This is the letter from Monty Cohen, who was director of the program at the time, admitting me to the medical physics program. And he felt it necessary to tell me that, I wasn't really maybe their first choice. He said, your admission here was touch and go. Your grades were borderline at the BSc program for admission to McGill. And so you're going to have to work really hard. I couldn't remember, so I had to actually go look back. I had a 79% average from UBC in the honors physics program in what was considered one of the top 3 programs. But I guess McGill standards were super high or something. So I came anyways. But as I learned, it was sort of expensive to live in Montreal. At least, it seemed like that. Tuition was only $500, but I lived at the corner of Penfield and Peel in a dumpy kind of 3-story walk up in a 1.5 apartment. And I had no scholarship. So after the first year, I actually had to interrupt my studies, and I couldn't start my project in the summer. I actually had to go back to UBC, live with my parents, and get a job to earn enough money to come back to the medical physics program. But, you know, as I said, lots of things are fortuitous in life, and this turned out to be a really important turning point. I started working with Meyer Bloom and Alex McKay in room 100 in the physics department on a project doing NMR and MRI of how water leaves wood when you dry it, which sounds a little obtuse, but I was working for Forintech Canada Corporation, but based in the physics department. And that's the Brooker NMR spectrometer I used back in the day. Because you had to dry wood, I actually had to build a gas furnace to be able to dry the wood. So I actually got a gas fitter license along the way, which is always something good to have. If, you know, your academic career doesn't work out, you've got something to fall back on. But that was really, you know, that was a turning point in so many ways, because Alex was a fabulous mentor, and he taught me a lot about NMR, as did Meyer. And, you know, even many, many years later, when I was inducted into the Royal Society of Canada, that work was cited, amongst other things, for my induction. So as a summer student, in between terms at the MPU, I actually did some of this really cool work. And I discovered the beauty of MRI. So when I came back, I did my second year project with Terry Peters in MRI on a 0.5 Tesla Philips scanner. And you could only get access. It was the only scanner in the province of Quebec at the time. And so you'd only get on the scanner after midnight, between midnight and 6 a.m. So this, of course, made me quite sleepy during my coursework during the day. So I took a lot of cat naps, and my roommate in Montreal took this picture. I think I was pulse programming for the Philips scanner and had fallen asleep at some point. But I still managed to get through my courses. The other thing that was really interesting about doing this, so the scanner was at the Montreal Neuro, was I started seeing and meeting neurosurgeons and becoming quite fascinated with how the brain worked. And I had an office in what was the brand new McConnell Pavilion at the time. And this elderly guy in green scrubs was always there at 5 o'clock in the morning when I had finished scanning. And I just kind of say hi to him, and he'd say hi to me. And after a couple of months of doing this, he said, well, I guess if we're going to share an office, we should introduce ourselves. He said, I'm Bill Findell. I used to be somebody. And I said, oh, hi, I'm Ravi Menon. And, you know, I didn't even know who he was. Turned out he was an extremely famous neurosurgeon, and he had just retired as director of the Montreal Neurological Institute. And they'd banished him to the basement, basically. So I learned a lot from him about the brain and the role that imaging could play in the brain, and got even more interested in that. So there really wasn't a PhD program in the MPU at the time. They were just kind of building it. And Bruce Pike, who was also a student in the program at the same time I was, I think he might have been, Noel Blais and him might have been the first two people to go into the physics program. Miguel didn't seem particularly keen to offer me a PhD position, but Terry Peters suggested I go work for Peter Allen, who I mentioned previously at the University of Alberta. So after driving east, I now drove west to Edmonton. I drove west the beginning of June. I was in White River on the Trans-Canada Highway. I don't think you can really see the mercury in that thermometer. It was minus 27 degrees on June 1st. So I wasn't too impressed. And it was snowing when I got to Edmonton. It's in June 3rd or 4th. So I really wasn't impressed. But what made it easier was I had straight A's pretty much in the MPU. And so I got an Alberta Heritage Foundation scholarship. That was $14,000, which was an absolute fortune in those days. Tuition at the University of Alberta was $1,000 for your first year and $50 a term afterwards. So you could have a fancy car and you could go skiing and you could do all sorts of things on that. So I had a great time in Alberta. These were some of my colleagues at the time. And this was when Peter Allen retired many years later. You'll recognize some of the people there who are members of the Medical Physics and Imaging panel at CIHR. So Christian Beaulieu standing on Pete's left, Nick Desanche, Rob Thompson, people like that, Alan Willman. So I had a great time there. And then Pete told me that, so I did my work mostly in myelination, in various models of myelination, in cats and crayfish and things like that. And it was suggested, well, now you should probably leave Canada and take a risk and go somewhere else and learn something new. So I applied to MRC, that was the predecessor of CIHR, and I applied to NSERC for postdocs, and they still didn't think I was worthy of scholarship of any sort. But Heritage Foundation funded me, and I had actually 14 postdoc offers from Harvard and Stanford and Oxford and GE Medical Systems and places like that. But this guy, Camille Ugerbel, intrigued me the most. So you remember Camille from that first slide. So he had moved from Columbia to the University of Minnesota and was building what would become the premier ultra-high field MRI facility on the planet. And that was kind of a dangerous move because he was building, this is a 4 Tesla magnet. It was the first such magnet ever built. It wasn't even installed at the time I committed to moving there. And a lot of people said this will never work. But I thought, you know, if this works, you know, I'll have a tool that nobody else has. And so he was so keen and so enthusiastic that I decided, you know, I'm kind of going to go to this somewhat unknown guy in this another cold place, you know, Montreal, Edmonton, now Minnesota, and learn something new. So I went there with the idea of actually doing cardiac spectroscopy, but almost as soon as I got there, Camille introduced me to Seiji Ogawa. Remember Seiji from that first slide? Seiji had just published a paper. So I moved there in 1990, and Seiji had just published a paper in which he described something called the BOLD effect, the blood oxygen level dependent effect, a way of making MRI images that were sensitive to the oxygenation state of blood vessels. And he predicted in his first paper that such images might be a way to supplant positron emission tomography to map regional oxygen consumption in the brain. So at that point, if you want to do brain function, you used either glucose PET or O15 water. And of course, both of those are slow techniques. Both of those involve some degree of radiation. So this is kind of a crazy idea, the idea that you could maybe use MRI to map brain function. MRI was only used for structural work at the time and was barely being used even for diffusion at the time. So Seiji came from Bell Labs every few weeks, and we started working on this. And in July 1991, in the lab in Minnesota, we were able to demonstrate convincingly to ourselves that This idea of using MRI to image brain function would actually work. And this particular subject that comes from a PNAS paper that we wrote on the subject comes from this young lady, Anne. And I mention her in passing again because serendipity, because I married her. So almost 30 years next year. She had one of the best brains that activated when we did these studies, as did the woman who Camille eventually married as well. So we both married our subjects, I guess. So fMRI was a big deal, functional MRI, which is, and so I had a lot of offers and I kind of wanted to come back to Canada, and I still had in my mind this idea of using MRI and medical physics and biophysics to help, people and patients. And there were 3 4 Tesla scanners at the time. They were all in the United States, one at NIH, one in Minnesota, and one at University of Alabama, Birmingham. None of them were connected to a hospital. And so I took the opportunity when it was presented to come to London, Ontario, and build a facility that was at the nexus of the university and the hospital and physically connected to both. So I moved to London in 1994. That was a big leap of faith because there was no building, no magnet, no nothing. We started construction in 1995, and my first 2 PhD students are in that picture. On my right is Chris Thomas, who's now a radiation physicist at Dalhousie. He was director of their residency program in medical physics for many years. And Brad Goodyear, who's a professor at the University of Calgary. So in 1996, we got our 4 Tesla scanner. So this was a different kind of scanner than the ones in the US. We had to do a lot of arm twisting. So the 3 in the US were built by Siemens, Philips, and GE respectively. They were big national, they cost tens of millions of dollars of federal funding in each of these places. And then people decided, you know what, this is just too hard, we're too expensive, and we're not going to do it. But I had the idea that we could make these things out of much more conventional technology using, conventional magnet cryostats and things like that. So I convinced Siemens and Varian, who had built the Minnesota system and then got divorced, to come back together and build something more commercially viable. So this was only a $4 million system instead of, maybe a $20 million system. So a little more practical. And this is what our center looks like these days. So this is what we built on the front lawn of University Hospital. And this is the Robarts Research Institute. So we're part of the university, but we're physically connected to the hospital so we could bring in patients. So that was my dream, and that's what we were able to accomplish here in London. I'll tell you more about what's in that building. So we've had a 7 Tesla human system since 2008. So that's about 11 years before Montreal got theirs. We've had a 3 Tesla Siemens Prisma also since 2008. It's been upgraded a number of times. And we have a 9.4 Tesla animal scanner that we do mice and marmosets and things like that. And next year we'll be installing a 15.2 Tesla mouse scanner. And the thing that makes us kind of unique also is the fact that, you all know that MRI is a unique imaging modality. And it allows lots of flexibility in terms of structural and functional assessments. And because it's non-invasive, you can use this to study normal brain development and healthy aging, from neonates all the way to, 95-year-old patients who may or may not have Alzheimer's. And you can study the evolution of development, of neuropsychiatric disorders, of neurodegenerative disorders, and so on. But the thing that's kind of unique about our center, and there are only like 1 or 2 centers in the world that have this, is that all our scanners are in one place. So we're very translational. And we cover species from mice to humans. And in most other places, including Montreal and Vancouver and Toronto and so on, you know, you have animal scanners in a vivarium or in one building, and you have human scanners in a different place, and they're all belong to different people, and there's very little cross-pollination between these. Here we have students who are working on a mouse project sitting next to somebody who's working on MS in humans, for example. And that kind of pollination, I think, is really, really important. So now let's talk about some science. I can't possibly tell you about all the things that we're doing here because it's a lot. I'm going to tell you about some stuff that's actually unpublished. It's brand new. It's literally a week old or so. So I'm going to tell you a little bit about how we utilize our 70 scanner and how I actually utilize some of the principles I learned at McGill in terms of using phantoms. It'll turn out to be a biological phantom to validate some of the techniques that we use. So I'm going to talk about the fact that we can use fMRI to bridge the mesoscopic scale, tell you a little bit about how fMRI works and vascular organization that underlies it. And we'll see how far we get here. So the mesoscopic, so there's microscopic scale, macroscopic scale, and mesoscopic. So normally we think of MRI as a macroscopic tool. Microscopic tools would be like, you know, the various microscopes, you know, light sheet microscopes, confocal microscopes, and so on. But these ultra high field systems actually allow you to bridge that. So the mesoscopic scale is really that transition from the microscopic, where you can study things that are very characteristic of an individual, to the larger macroscopic scale, where you're kind of doing averaging, you're coming to conclusions about species typical anatomy, for example. And, you know, Montreal is very famous for this. Everybody uses the M&I template, for example, for brains. That's a macroscopic, averaged across many, many people reference atlas. So this is the scale at which fiber tracts connect different brain regions. Fiber tracts, of course, we can image with diffusion tensor imaging. And it's arguably somewhere in the neighborhood of tens of microns to centimeters. In mice, if you can, this is a coronal section of a mouse brain. This is histology on this side, and this is atlas, the Allen Brain Atlas on the other side. You see the cortex has, you know, 6 layers. It has different parcellations. You know, these parcellations are of the order of, you know, a millimeter or 2 in size, and the layers are a few hundreds of microns. But we can image that at 9.4 Tesla and 15 Tesla. So functional MRI relies on neurovascular coupling to provide an indirect measure of what neuronal ensembles are doing. So we're not actually measuring brain function, we're measuring the hemodynamic response to brain function. But it works across species, and it allows us to bridge between animal models of normal function or disease to humans who are normal or who have some form of disease. Give you a sense of what that looks like. You know, in a mouse, you know, sort of state of the art, we can image an fMRI signal from, you know, something like, you know, 200 by 200 by 200 microns, give or take. That has about 25,000 neurons in it, in a mouse. In humans at 7 Tesla, we typically image just submillimeter. So 0.8 by 0.8 by 0.8 is pretty much state of the art. So that has about 700,000 neurons in it. So, we're not microscopic, that's for sure. And at 3 Tesla, of which there are, hundreds of machines even in Canada these days, we typically image at, 2 by 2 by 2 or 2.5 by 2.5 by 2.5. And so there are about 21 million neurons in those, in a single voxel in a 3T image. So it's important to be able to look at these scales of activity. So how does fMRI work? Well, fMRI works because of the difference between paramagnetism and diamagnetism. And I'll come back to that in a second. The paramagnetic materials are those that enhance the local magnetic field. So if I put a paramagnetic substance into a bath of water in an MRI scanner, it will locally enhance the magnetic field there. Diamagnetic materials actually try to oppose that magnetic field. Okay, so if I put a diamagnetic substance into a homogeneous bath of water, put it in an MRI scanner, you will actually see that it reduces the field. Why is this important? Well, let's go back to Linus Pauling. Remember, he was the start of my serendipity slide. So he published a paper back in 1936 on the magnetic properties of hemoglobin. Hemoglobin, as you know, is the oxygen carrying unit in your red blood cells. Hemoglobin has 4 heme groups. Each heme group contains an Fe2 plus ion, and each of those ions has 6 coordination sites for oxygen. Well, for various things, 5 of which are occupied and 1 of which binds oxygen. So the cool thing about hemoglobin is when it is fully saturated, so it's gone through your lungs and you're not a smoker, your blood is fully oxygenated, you have 4 O2 molecules bound, and oxyhemoglobin, as we call it, is diamagnetic. The second you start plucking off 1 or more oxygen molecules, it becomes paramagnetic. So the magnetism of the hemoglobin molecule changes depending upon its oxygenation state. That is really the basis of fMRI in a nutshell. So let's look at this from a vascular tree perspective. So we have oxygenated blood coming in from the arteries, coming from your heart and your lungs, and it's diamagnetic. And so we can call the field shift 0 parts per million because water is also diamagnetic. And so you can't really distinguish using magnetic field maps or a technique that's sensitive to magnetic fields. You can't really distinguish an artery from anything else in the brain. The venous side, however, is now draining blood that's coming from capillaries, and that blood is partially deoxygenated. Not a lot, you've lost about 30% of the oxygenation. So it's deoxygenated and it's paramagnetic, and there's a small magnetic field shift. So, oops. So in between those 2 pieces of the vascular tree are, of course, neurons and astrocytes. So this is the business end of the brain, very important for brain function, obviously. And they get their energy, the glucose, their oxygen, and the waste products are drained by capillaries. And the capillaries are where most of the oxygen and glucose exchange actually occurs. And if you use the right kind of imaging sequence, which is a susceptibility weighted sequence of some sort, typically a gradient echo sequence, you can actually image this difference. And this is probably one of the most beautiful experiments ever done in MRI. This was done by Seiji Ogawa at Bell Labs with a rat in a 7 Tesla animal scanner, in which he first imaged the animal breathing 20% CO2, regular air. Oh, the rest of it was regular air, so lots of oxygen and some nitrogen. And this is the kind of contrast he got, not very exciting. You can see the corpus callosum here. You can see the cortex. You can maybe see the hippocampus. Not great. But then he switched it to normoxic conditions. Under normoxic conditions, you start to deoxygenate some of the venous draining vessels. And this is what happened. He got an image in which the contrast changed based on the oxygenation state of the brain. It's a simple experiment, but nobody ever thought to do it. And it was brilliant. It led to the discovery of functional MRI. Interesting thing is, he chose not to patent this. So he convinced the president of Bell Labs to not patent it because this would be a benefit to mankind. So Bell Labs did not patent this. But of course, nobody cares about rat brains, and that's why he was coming to Minnesota, where I was a postdoc, to try to demonstrate this in humans. So this is what the bold response looks like in real life. So these colors actually mean something, because if you look at the surface of the brain or look into a brain, this is what you see. Oxygenated blood is red, deoxygenated blood is blue, and the capillaries are somewhere in between. So if essentially you do an optical spectroscopy experiment, which is what your eyes are doing, you can actually tell the oxygenation state of the tissue by looking at the color of the blood vessels. Now, we actually wasted a lot of time in Minnesota initially, because we were looking for a signal that went down. So you would think that if you do a task and the neurons become more active, right? So your active neurons, they consume more oxygen, they consume more glucose. They would use, that oxygen has to come from somewhere. So it comes out of the red blood cells, and that would give rise to even more deoxyhemoglobin. So the blood would get bluer, or the MRI signal would get darker. But that's actually not what happens. And this confounded us for quite a while. What happens is the blood flow in an area of heightened metabolic activity goes up dramatically. So you actually have more blood flow and more oxygen than is actually required by the tissue. This is an interesting aspect of biophysics. It's almost as if you have to increase the gradient, the oxygen tension gradient, between the red blood cells and the mitochondria in the neurons. And so flow typically goes up by 50%, but oxygen consumption actually only goes up by maybe 5%. And so the net result is the signal actually goes in the opposite way. The deoxygenated blood at rest becomes more oxygenated, and the signal goes up. And if anybody had read Wilder Penfield's notes when he was at the MNI, they would have figured this out, because Penfield noticed this in the 1960s when he was operating on people with epilepsy and they had seizures while they were on the operating table. He noticed that their blood got redder, not bluer. And he wrote in his notes that the brain blushes. So, but we weren't aware of that at the time. So we were looking for the signal going in the wrong direction. Eventually we figured it out, of course. And so Seiji actually has won many prizes. He won the Gardner Prize in 2003, and I was fortunate to be invited to that event. Actually, in that picture of the 5 laureates, he's actually the only one who has not won a Nobel Prize in that group, which is quite interesting. So the vascular organization of the brain also works to our advantage because blood vessels are everywhere in the brain, and they're found at all kinds of different levels in terms of layers and columns. And the vascular density turns out to be proportional to synaptic density. So Mother Nature has evolved to put lots of capillaries in areas that are very metabolically active. So that all works to our advantage. But there's some things that don't work to our advantage. So I showed the map this way, but that's not the way the brain's actually organized. What you have is arterial vessels that come into the gray matter from the surface of the brain. They go through capillaries, and then they come back up to the surface of the brain through veins. So this is gray matter, and this is white matter. And if you look at it from, look at an actual picture, this is what it looks like, the surface of the brain. So some of these are arteries and some of these are veins, but there are vessels everywhere. This is a 2 millimeter scale bar. So you can see that there are relatively big vessels. These are not capillaries, but they are located, every couple of 100 microns or so. You can blow that up. There's a single artery, and these are what we call penetrating arterioles that go into the brain. And then we have these things, draining veins that come out. So when you image at 3 Tesla, or you image with low resolution, say 2 by 2 by 2 millimeters, several interesting things happen. The blood volume in the brain is dominated by vessels that are bigger than 100 microns. So most of the blood is in vessels that are bigger than 100 microns. And most of those vessels are actually on the surface of the brain, and some of them are penetrating. Which vessels we image depends a lot on what field strength we use and what kind of imaging technique we use. But the problem is draining veins are always a problem no matter what. So this is work. This is a landmark paper done by Joe Gaddy, who was a master's student working for a colleague of mine, Brian Rutt, here in London. He's been our managing director for our imaging facility for 28 years now. He did an experiment where he imaged the same people at half T, 1.5 T, and 4 T at the time. And you can actually do fMRI at half T, but you don't have to know a lot about the physiology of the brain to realize these are venous vessels. What you're imaging are big draining veins. When you go to 1.5 T, you still see those draining veins, but you start to see some other stuff. And when you go to 4 T, well, the veins are still there. They're really bright. But now a lot of the gray matter, the tissue signal, is showing up. So at low resolution, you're imaging all of that. You're imaging the peel vessels, you're imaging these penetrating vessels. The voxel contains everything. And this is what the bold signal looks like, at something like 3 Tesla. You get about a 2% signal change. But remember, this is a hemodynamic effect, and the hemodynamics are sluggish. So if you give a brief 2-second visual stimulus, for example, you don't see a response in the visual cortex for several seconds, and it takes several more seconds for it to peak. So fMRI is relatively slow, much faster than PET, of course, but very slow compared to EEG or something like that. So a lot of work in my lab is aimed at pushing the spatial resolution of the technique. And nature provides us with 2 ways to verify how this is working. So one is to examine something called cortical columns. So some of the brain, maybe all of the brain, but we don't even know yet, is organized into small areas of specialization called cortical columns, which they run perpendicular to the surface of the brain, typically maybe 1/2 millimeter for the square here and 3 millimeters deep. Or maybe 1 by 1 by 1 millimeters. And it varies depending upon whether you're in auditory cortex or visual cortex or motor cortex. But if you can figure out ways to activate these individual columns, then you have an in vivo resolution phantom. So why is this important? Well, we can test columnar specificity. Going back to my model here, if you can activate a cortical column, so I'm showing here only some neurons being activated, say 1 millimeter apart, you would get capillary changes, it turns out, that are very tightly coupled to that. And we know this from optical imaging with microscopes. The problem is those drain into these annoying big draining veins, and they are distal to these columns, and they collect blood from non-activated and activated columns. And this is where a lot of fMRI is really sensitive to. So if you're only sensitive to these vessels, you can't distinguish columns. But if you were sensitive to capillaries, you could. So that's a key in vivo resolution phantom approach. So one such set of columns are ocular dominance columns. So in your visual cortex, you can activate left eye columns by stimulating only the left eye and right eye columns by stimulating only the right eye. So this turns out to be very simple. This has been done in monkeys using carbon-14 deoxyglucose. And it's also been done in monkeys using tritiated proline. But it had never been done in humans. And my student Brad Goodyear, who I mentioned earlier, he first demonstrated this at 4 Tesla. Kang Cheng at Riken in Japan did actually much nicer work in 2001. And then people have been using this for various purposes ever since. Works beautifully at 7 Tesla. So that's one organizational layer. The other is cortical layers. So the cortex is defined or sort of delineated into 6 neural layers in humans, which I've marked off schematically here. And these layers do different things. And so if you could image at the level of layers, you could actually try to understand some of the inputs and outputs into the cortex, which we have no tools to be able to do in humans currently. So this is a single sulcus. So here's the sulcus in the visual cortex. It's got a cortical vein. Remember, this is the surface of the brain. It's in an invagination in the cortex. And this is what the vasculature looks like. So this is gray matter. You can see gray matter has a high degree of capillary and other vasculature compared to white matter, which is further away. And if you blow that up, this is what it looks like. There's 6 neural layers, and there are, for some unknown reason, there are 4 vascular layers. And as I mentioned, if you can image layers, you can actually look at interactions, because the inputs into the visual cortex come into layer 4, and they are segregated. They go left, right, left, right, left, right. When you look above, they start to interact. This is how information between the eyes is exchanged. So how do we do this in humans? I'll just spend a couple of minutes on this because I've got a few extra slides, but we won't do that. So this is my life. We design RF coils a lot. So this is a 7 Tesla coil designed specifically to look at the human visual cortex. It uses 8 channels for transmit and 32 channels for receive. We have an RF group that currently has Ph.D. Kyle Gilbert, and a electronics technician, and we have another engineering person we're hiring. So we have to design these and make sure they're safe for humans. So that involves SAR, specific absorption ratio modeling, mapping the magnetic transmission fields, and so forth. But basically, the person lies in this, they look at a mirror, we project onto a screen, and we can project whatever we want onto that screen using MRI compatible screens. Compared to a head coil, we're able to double or triple the signal to noise ratio. So that's very important. We control which eye looks at the stimulus using LCD goggles that are, again, MRI compatible. This is work that Brett Liam, a student who came to me from Acadia University, and Atina Akbari, who's a postdoc in my lab, who came from Queensland, Australia. They've been doing this with simple stimuli, gray background for 30 seconds, both eyes for 30 seconds, gray right eye, left eye, and this coil. And I'll end with this last slide here, I think, which is just one result slide. We also do a lot of MRI physics to try to understand different ways to be sensitive to the capillary signal as opposed to all of the vasculature or all of the venous vasculature. There's a technique called VASO that was developed not by us, but we use it, which is sensitive to blood volume changes only in the capillaries. Its problem is it's slower than the bold signal, and its sensitivity is lower than the bold signal. But if you look across the layer, so this is the surface of the brain, and this is the white matter, and this is the signal across the layers, you see that VASO gives you a very different result than bold. Bold gives you high signal changes at the surface. VASO gives you high signal changes in the cortex. And that's because of their differential sensitivity. VASO is sensitive to the capillaries. Bold is sensitive to the capillaries, but it's also sensitive to all this other nonsense that we're not interested in. So, many of my CHR grants and things are all spent on this kind of stuff, as well as applications in humans. We've done some work in radiation biology over the years, trying to use actually the bold effect to discriminate tumor recurrence from radiation necrosis, for example, and we've published on that. It actually does work. But I'm not going to talk about all of that. I'll just thank all the funding agencies who've supported this over the years, and stop sharing at this point.